and our last talk is from Koji Yahara from Japan. So he has put a presentation together as a pre-record, and so I will share that now. And then all of our panel members will be ready for questions afterwards. And I can already see that there's Discord discussions and questions going on.
I'd like to thank Ben, Nicole, and organizers for giving me this opportunity to share and discuss a problem I'm facing toward incorporation of genome sequences into Campylobacter AMR surveillance. This slide shows a Campylobacter AMR surveillance so far in Japan, Tokyo. For human diarrhea cases, percentage of resistant strains for erythromycin, nalidixic acid, and fluoroquinolones is annually measured for C. jejuni and C. coli, respectively. Clearly, the percentage of resistant strains for erythromycin and nalidixic acid is high in both species. The number of annually tested strains is approximately 100 for C. jejuni and 10 for C. coli. So obviously, this is a small-scale surveillance, and the data are phenotypic, measured using disk diffusion method. Similar phenotypic surveillance is conducted in farms. For C. jejuni strains isolated from healthy cattle in farms, approximately from 40 to 70 strains were annually tested, and percentage of resistant strains was annually plotted for each priority antibiotic. Notably, the percentage of resistant strains was high in tetracycline, as well as fluoroquinolone, ciprofloxacin, and nalidixic acid, whereas the percentage of resistant strains was low in erythromycin, which are similar to the human diarrhea cases. For C. jejuni strains isolated from healthy broilers in farms, overall resistance pattern was similar to the strains from cattle, whereas they also showed a high resistance in ampicillin, on the contrary to the strains from cattle. Similar phenotypic surveillance is also conducted in slaughterhouses. For C. jejuni strains isolated from chickens in slaughterhouses, the resistance pattern was similar to the strains from broilers in farms. These surveillance data are publicly available in One Health platform website with other MR surveillance data in Japan. For example, all routine bacterial culturing and antimicrobial susceptibility testing results across more than 2,000 hospitals. As I mentioned, the data so far were only phenotypic and measured using disk diffusion method. Very recently, our team started to consider genomic surveillance, in which we annually obtain genome sequences as well as the phenotypic data of 100 human diarrhea cases, and 100 chickens, and 100 cattle, broilers, and pigs in the farm. Then a problem I would like to discuss with you is what is the format of internationally meaningful annual report, and what is an advantage of whole genome sequence in this context. My current idea is an essential component is core genome tree with MLST and resistance patterns, and the presence of absence of specific genetic features, as shown in this figure of Neisseria gonorrhea genomic surveillance I promoted. In order to do so, we have to list up specific genetic features underlying the resistance to each priority antibiotic. Although such genetic features would be unclear in Campylobacter, except for the quinolone resistance determining region. Furthermore, it would be nice if we could add information of specific virulence factors to be monitored. Although I heard from Simon and Ben that Campylobacter's virulence is not attributable to a specific gene. Frequency distribution of sequence types is easy to create and is internationally comparable. So this kind of figure will be a component of the report. A more advanced component would be a figure that examines association between presence of specific genetic features and the extent of resistance to an antibiotic. Furthermore, if possible, it will be beneficial to integrate with other countries' data so that international comparison is included in this report. I appreciate any comment and discussion from you. Thank you very much for your attention.
Okay, thank you to our speakers. So do we have our speakers? If you could perhaps either turn mic on, or if you're willing to, your video. I'm going to start the questions by dealing with specific questions that have come on through this Discord or other means. So, sorry, I'm just on two screens here. Okay, so we go back to Frida and Amy, and there were a couple of questions from Ozan. Do you think the increase was due to people not going to the GP or hospital to report, or people more careful with the increased health and safety measures after the pandemic started?
Frida, shall I take this one?
Absolutely.
Thanks. So it's really challenging to try and disentangle the different drivers. It's going to be multifactorial. I think we saw for other pathogens like Salmonella, so we have routine whole genome sequencing for the majority of Salmonella isolates in the UK. And what we saw with the clusters we were detecting routinely was that during the lockdowns, the cases we were identifying were generally rather old or rather young, and they had more severe clinical presentations of illness and more likely to be hospitalized. I expect we would probably see a similar picture for Campylobacter, but because we don't have the same level of whole genome sequencing or routine questionnaire data, it's really hard to try and understand the drivers. But what is quite striking is that once the by sort of winter time, the laboratory reports kind of recovered to levels that we would normally see, whereas that didn't happen for a lot of other pathogens.
Yeah, okay, so I think you've, and others have sort of followed up on Ozan's other question, which is, would you say this is a longer term trend with the pandemic leading to increased public health awareness? So I think you've all actually covered that, unless you want to say any more, Amy. Okay, so a question to Amy and Frida. What is the minimum and maximum number of cases in each Campy outbreak? Do you have that granularity of data to your hand, Frida?
Amy may.
So I don't, but I know for outbreaks this year, so one of the ones linked to chicken liver parfait was over 60 cases. So some of them can be, because we don't have all these outbreaks are dealt with at the local level, there has to be a really strong epidemiological signal. So that would be either reports of illness after attending an event like a wedding or a christening, or it might be linked to a specific producer, like we've had problems with raw milk outbreak, Campylobacter outbreaks associated with raw milk. So it would be very unlikely that we would identify an outbreak of probably less than 10 cases. But there was a project done a couple of years ago that did sentinel surveillance using whole genome sequencing and structured questionnaires for 2 parts of the UK. And through that, we were able to identify smaller outbreaks based on the genomic comparisons, MLSD, and also our own in-house SNP typing. But routinely, they're probably on the larger side, I would say.
Thank you. There's some questions in for and so Nabil asks, how do you explain the decrease of Campylobacteriosis in Canada? Opposite trends are found in many industrialized countries, especially due to syndromic PCR that facilitates the detection of Campy in human stores compared to culture alone.
Okay. I can hope that it's due to public health measures to decrease. Sorry, can you hear me?
Yes, we can hear you.
Can you hear me?
Yes, we can hear you.
Hello? Okay. Okay, we'll hope that lasts. Maybe I'll turn my video off just to try and help with the internet. We can hope that that's due to public health measures that were designed to decrease Campylobacter in human cases. At this point in time, the Canadian Notifiable Disease Surveillance System also is only using laboratory confirmed isolates and not the PCR that was mentioned in the question. So whether that has something to do with it or not, but at this point, it's still laboratory confirmed cases. Isolates.
Okay, thank you. I think we got the end of that reply.
Can you still hear me?
Yes, we can still hear you. So there was a question from Fran but I think Anna's answered that in the Discord. So it relates to some question regarding ciprofloxacin resistance in chickens and turkeys. I'm going to move on to a question for Koji. So this is from Desi. Do you see any correlation between restaurants that offer raw chicken dishes and Campylobacter cases? Which
I have
think you
already
just
answered in Discord. Sorry, I don't have the data to answer, to explore the correlation.
Okay, thank you. Sorry, I'm operating off two screens and trying to fill the questions at the same time. So a question from Nicole, which is to everybody. Why do you suspect fluoroquinolone resistance is increasing? Is it selective pressure or are there other factors in play? Anybody want to take that one?
I attempted to write something in the chat, but I think
Sorry for it, Hans.
there's no doubt that selective pressure will have an impact on, it's so easily selected for, if you look at what happens in a chicken flock when you treat them with a fluoroquinolone, you know, you select for fluoroquinolone resistant Campylobacters just like that. However, if you're then reducing the usage in the industry and it's not going away, my suspicion is that it's not costing Campylobacters an awful lot to bear that resistance, and that may be why, even though selective pressure goes away, you don't see a concomitant, you know, proportional decrease in the proportion of resistant organisms. But I must admit, personally, I think it's because you're selecting for resistant organisms. That's why it increases in the first place.
Thanks, Rita. I mean, I guess anybody else could jump in. I'm trying to recall, I remember some papers 10, 15 years ago that looked perhaps in vitro, although there might have been a follow-up study in chickens that actually fluoroquinolone resistant Campylobacter, even in the absence of selection, were more fit, both in vitro and in vivo. So I'm afraid I don't recall the author of the paper, but
Andrew, I posted a paper on the Discord from Tycho Cronin's group where they actually showed that fluoroquinolone resistant ones are more virulent and are better at biofilm formation. They published that last year. It's all due to changes in supercoiling that the gyrase is involved with. So they are probably at least as fit or more fit in certain environments.
Thanks, Arno. Right, and I'm coming in on email as well. So in this question from Nicole, actually, in Canada, it looks like the recovery of Campy at retail is going down, while in the UK, it's 40%. And if you're still with us, do you want to comment on that?
So I think that was almost partially answered previously, that there might be different intervention methods or different ways of looking at it in Canada versus in the UK. So we can continue that discussion further on in Discord, and likewise the situation that's happening in Japan. We're starting to come close to the end of the session. So Andrew, how many more questions do we have for the audience or for the panel?
Well, I think we're about there. Actually, there isn't anything else that's come in, and I've now done Zoom, Discord, and email, and I think we're covered.
It's fabulous. We are very integrated in our platforms today, and so I think it's coming to the end of the session. So I have a couple of thank yous specifically to all of the speakers and our public health invited speakers as well. It's been a very packed 3 hours. So thank you everyone for your patience and all of the discussion, both in the live sessions and on Discord. We'll start more Discord interactions can still continue after this session. And you can follow up with people as you wish after that. I'd also like to thank the co-chair, my co-chair, Andrew, and Lisa and Ricardo, who have been milling in the background as well during the session. So with that, let's do a little preview of tomorrow. We have tomorrow as our last day of talks. We have a session on survival and application in the morning, followed by the industry panel. And then following that, the plenary with some invited guest speakers and guest panelists. So you can ask questions to the panelists at that point. And then finally, we are closing the conference off with Campy quiz. So Campy quiz night for our Friday. And that's to kind of stretch your trivia muscle and socialize a little bit more on our Discord platform. So it's been a great pleasure to host this session. And I want to again, thank you everyone for attending. And with that, we will close the session and see you on Discord.